Assalamu alaikum everyone. I'm Abdulrazzaq Qattea and I'm a third-year medical student. Just before we start, I need you guys to, if you have any question or any concern, just unmute yourself and feel free to speak. And if you have any question regarding anything or for the exam question, you can approach me by WhatsApp. I will be more than happy to help you. Okay, pancreatic secretions. In order to understand and enjoy the lecture, I need you guys to understand some concepts, which is what's the goals of the pancreatic secretions. We have two main goals. The first one is we need to remove the acidity after the stomach. So the stomach is known for being acidic environment, and it has a lot of enzymes that is acidic. And if these enzymes go to the duodenum and small intestine, it will harm them and cause ulcers. So in order to compensate for that and neutralize the acidic enzymes that is going to the duodenum and small intestine, we need a base. And this base is called bicarbonate. So the first goal is to neutralize the acidity by bicarbonate. The second goal for pancreatic secretions is we need to break down the food. So the pancreas will have a lot of enzymes that will break down the food, and it will break the three main sources of the energy, which is proteins, fats, and carbohydrates. The other thing is the pancreas is known to be an exocrine gland and endocrine gland. What does exocrine mean? It means the pancreas will secrete its content and its enzyme out of the blood vessel. Okay. So the focus of today's lecture is to understand what's the exocrine as the pancreatic enzyme in the exocrine gland. However, in the endocrine gland, which is the pancreas, it will release the hormones into the bloodstream, which is insulin and glucagon. You will study the endocrine in the endorepro, but for the exocrine, we will study it now. In order also to understand the physiology, we will discuss a bit of the anatomy as well. The exocrine gland will have two cells, or two main cells. The first one is the duct cells, and the second cell is the acinar cells. And each one of them will have its own function and its own enzymes and secretions. Okay, so if you look at this picture is a picture of the exocrine gland. The exocrine gland will have acinus and ductal cells. If you have a look at the green part, which is the acinar cells, which will be at the beginning of the gland, this is called acinar cells. And what's the function of these cells? They will secrete the digestive enzyme, which is amylase, trypsin, and so on. Another thing is they will secrete the sodium and chloride, and they will control the isotonicity. Okay? So what's the function of the acinar cells? Is to secrete the digestive enzyme. And if you have a look at the red cells, they are the ductal cells. And what the ductal cells do, they will not do this as same as the acinus. They will secrete the base, which we talked about, which is called the bicarbonate or the hydrogen carbonate. There's no difference in both. I saw Dr. Abdul-Jabbar talking about the hydrogen carbonate, but I will be mainly talking about the bicarbonate. There's no difference. طيب. What happens is when the acinus secretes its enzyme and everything, the bicarbonate will come from the ducts, and it will also modify everything in the duct also. So another function of the ductal cells is to modify the fluids. And if you have a look at this picture down, this is the pancreas. So this is the exocrine gland here, and then we will have the pancreatic duct. and the pancreatic duct will empty into an opening called the ampulla of water. And this ampulla of water have a sphincter. Sphincter is basically a muscle. It's controlled by a smooth muscle, so it will open and close depending on the need and secretions. I just need you to focus on something, which is, let's look at the gallbladder. The gallbladder will have the common bile duct, which will share the same opening for the pancreatic duct. So both the gallbladder and pancreatic duct, they will both empty into the sphincter of body or ampulla of water. And this is important later. Just memorize it now. Like, let's now focus on the ductal cells. What did we say? What's the function of the ductal cells, guys? What's the main function of the ductal cells?
Release bicarbonate and modifying fluid, which is like maintaining their isotonicity.
Yes, exactly. So the ductal cells' main function, as our colleague said, to release bicarbonate and modify the fluid. Like, bicarbonate have two functions. The first one, as we said, when we have hydrochloric acid and the chyme after the stomach, it will go to the duodenum and small intestine. And the acidity will harm this structure, which is the duodenum and small intestine, and it will cause something called peptic ulcer or ulcers. I will show you what's ulcer in the next slide, but now the function of the bicarbonate is to neutralize these acids, which is coming from the stomach. Just a second. I will play. The second function, after neutralizing everything, do you guys remember when the doctors talked about the hydrochloric acid and the pepsin as being is functional when the environment is acidic? This for the pancreas, it's the opposite. So the pancreas or the pancreatic enzymes will work on more alkaline environment. So the first function of the bicarbonate is to neutralize the acidity and then activate the pancreatic enzymes, which work on the more alkaline environment. Now, this is an ulcer. This is a biopsy of a small intestine. So as you can see here, ulcer means there is a separation in the epithelium, which is caused by an acid. So this is small intestine biopsy. It should be continuous. But when the acid came, it will degrade and break down everything on the way. So it will cause a gap. So this gap is called an ulcer. So what's the main stimulus for bicarbonate? And how can the pancreas secrete the bicarbonate, and when does it should know that the body needs to the pancreas needs to secrete bicarbonate? Simply by acidity. صح? It makes sense because we only secrete bicarbonate in order to neutralize the acid that comes from the stomach. So the stimulus for bicarbonate is acidity in the duodenum. So what happens is that after the stomach empties into the duodenum, there is a cell called S cell. This S cell is going to sense the acidity or the dropping in the pH, and then it will release an enzyme called secretin. So what secretin function is it will go to the ductal cells and then secrete the bicarbonate. And why ductal cells? Because as we said before, the ductal cells is responsible for bicarbonate secretion. Now, unfortunately, the secretin will not activate it directly. We need a cascade. What does cascade mean? It means, there is more than one steps in order to activate the channel. What secretin do is it will go and open the CFTR channel, which is a chloride channel, and with the chloride channel, the bicarbonate will go with the chloride and be secreted out. And this is by a signaling, which is cyclic AMP. Signaling means more than one steps or a cascade, which is more than, just don't worry about this. We will talk about this later, inshallah. Just know secretin will secrete bicarbonate by opening the CFTR channel and by cyclic AMP. طيب, this is important slide. طيب. Bicarbonate have two sources in the ductal cells and have two mechanisms for secretion. Secretion where? Into the duodenum. تمام? So just a minute. طيب. We have two sources and we have two types of secretion. Remember this, okay? Doctor might ask you about this slide. So what's the first source? First source is called the carbonic anhydrase. So what's carbonic anhydrase simply is, we have a water and we have a carbon dioxide. And then when they came with each other, they will form one acid and one base. This is called carbonic anhydrase. So for the acid or the hydrogen, as we said, we don't need the environment and the duodenum to be more acidic. So what we're gonna do with the H or the hydrogen is we're gonna kick it out of the cell. We're gonna kick it to the interstitium. By what? By a channel called sodium hydrogen exchanger. So the hydrogen will go outside and the sodium will go outside. This is for the hydrogen. And for the bicarbonate, as we said, this is the first source, which is the carbonic anhydrase. The second source is called is by sodium bicarbonate cotransporter. What does cotransporter mean? It means we have two substances or two ions that will go to the same direction. So the sodium will go inside and the bicarbonate will go inside. And then we will have a bicarbonate inside the ductal cells. This is the ductal cells. This is the interstitium, and this is the duct lumen or the pancreatic duct, or we can say this is the duodenum here. طيب. These are the two sources. First one is carbonic anhydrase, and then we have the sodium bicarbonate cotransporter. طيب, how we after having a bicarbonate, how can we secrete it out of the cell? We have also two types of secretions or two mechanisms of secretions. The first one is the anionic exchanger. So what is an ionic exchanger? Is that after the bicarbonate being in the cell, it will go out and it will bring in a chloride, which is exchanging of bicarbonate with the chloride. So how can you remember this? Is by look at the chloride, it's negative, and look at the bicarbonate, it's also negative. So basically, you don't want a lot of negatives to be on the same side. So what you're gonna do is you're gonna get the chloride inside the cell and the bicarbonate outside the cell in order to balance the gradient. And the same thing for here for the sodium and bicarbonate. Bicarbonate is negative and sodium is positive. So you want a balance. So you will get them both inside the ductal cells. The second, this was the first secretion of bicarbonate, and the second one is by the CFTR channel. Let's go back to the S cells. Do you guys remember what S cells do? S cells do. Yes, exactly. So it will release secretin. So look here at the right side. If after the secretin goes to the ductal cells, it will bind and it will activate the adenylyl cyclase. What adenylyl cyclase do? It will convert ATP to cyclic AMP. Remember the cyclic AMP from the secretin, okay? So after cyclic AMP is being activated, it will go and activate the protein kinase. What protein kinase do is it will go to the CFTR channel and it will activate it. So when we open the CFTR channel, we're opening a channel for chloride and for bicarbonate. So what we're gonna have here after opening the CFTR, we will have secretion of both bicarbonate and a chloride. But notice here the chloride will go back to the cell by the anionic exchanger. So it's a cycle where the chloride will go inside always. Okay, we're done with the secretion. We said there's CFTR and there is an ionic exchanger. Is this concept clear? Okay, great. I guess no questions. Type. For the bicarbonate, there is something called effect of rate. Okay, great. There's something called effect of rate. So what's effect of rate is simply when a higher rate of secretin means more stimulus, means we need more bicarbonate. So what does this mean is when you eat food, there will be a secretin stimulation. And when we have a lot of secretin, we will open the CFTR channel and we will have a lot of bicarbonate. That makes sense because when you eat, you will have more food in your stomach, and more food in your stomach, more acidity. So the more acidity will go to the duodenum, you will secrete more bicarbonate. So when you secrete more bicarbonate by secretin, you should expect that the chloride will go down. And why is that? Because remember, let's go back one slide, remember this channel, which is an ionic exchanger. It will secrete a bicarbonate and will get a chloride inside again. So the chloride will be low and the bicarbonate will be high. This is in case of a higher rate of secretion. Okay, but in case of low rate of secretion, the opposite will happen. The chloride will go up and the bicarbonate will go down because there is no food, so there is no secretin, so there is no bicarbonate going up, and thus there is no anionic exchanging. And this, just memorize this, which is low flow rate will have more chloride and high flow rate more bicarbonate. Simply يعني low secretin or high secretin. You can remember it by this. Okay, another concept here you have to understand. So simply this graph is telling you about the fluid which comes from the acinar cells until it goes to the ductal cells. So the blue box here is acinar cells, and the orange or the yellow one is the ductal cells. Okay. One thing you need to understand before we go into this diagram is that the acinar cells will have isotonic secretion. What do I mean by isotonic secretion is the acinar secretions will have the same count of ions as the plasma. So isotonicity means the fluid of the acinars will have the same concentration of ions compared to the plasma. So in the plasma, we have more sodium or more potassium in the outside environment of the cells يعني. What do we have more? Is it sodium or potassium? Yeah, guys, you can say anything. Yes, it's a 50-50. Yes, so it's sodium. So look here at the sodium, it will be higher than the potassium here. Another thing to notice is that the potassium, the sodium, is not changing, and the potassium is the same thing. It's not changing. Why is that? Because when the acinar cells is secreting the potassium and sodium, it's going to the ductal cells, and there is no channels that can exchange the sodium and potassium. So they will stay the same. They will not be affected. However, if you looked at the bicarbonate, you will notice that it's getting more and more, and it's getting higher when it goes to the acinar cells, to the ductal cells. And why is that? Because simply the ductal cells is secreting more bicarbonate. And when we said that if we have more bicarbonate, the chloride will get low because of the anionic exchanger. Okay? So when you look again at the bicarbonate, you will see that it's becoming higher and higher. At the same time, the pH is getting higher and higher, and it's getting more it's leaning to the base pH. So rather than memorizing, just try to understand this concept. Okay, guys. This is a checkpoint. Do you have any question for anything previously? Or should we move on? Okay, thank you. We're done with the ductal cells. Just a small recap. We said that it's a ductal cells that secretes bicarbonate, and the bicarbonate is being secreted by the bicarbonate being secreted by the S cells, and it will go stimulate by secretion. The second one is the acinar cells, and the function of the acinar cells is to release pancreatic enzymes to digest the food. So what are the enzymes that is being secreted from the acinar cells in order to digest the food? Just know that the pancreas will digest and break down and degrade the three main source of energy, which is carbohydrates, proteins, and lipids. For the carbohydrates, it will release amylase, and for protein, it will release DOS, and for lipids, it will release lipase and phospholipase. Why did I go through it rapidly? Because the doctor did not pay attention to any of this. So just know them, yani, in case. Now, the pancreas will release those previous enzymes in inactive form. What do you mean by inactive form? It means these enzymes cannot digest and break down anything because they are inactive, and they are precursors for the active enzyme. So we have the inactive enzymes. How can we know these are inactive and these are active? Simply by looking at the name of the enzyme. If the name have a gene, or it's called trypsinogen, or chymotrypsinogen, gene means a precursor, or it's not active yet. The same thing for broelastase. When you see a bro, this is inactive. The same thing for these two enzymes. So when we remove them, It means these enzymes are active and ready to destroy and degrade and break down the food. Now, what's the point of secreting these enzymes in inactive form? Simply because the pancreas can digest itself. So we need these enzymes to be inactivated because the pancreas is mostly lipid. So when we have a lipase and this lipase is eating the pancreas, there will be something called pancreatitis. We will come to this concept, but just know that the inactive form will be active if we have a trypsin. And the trypsin will be, if we have a trypsinogen converted to trypsin by enteropeptidase. Okay. So what are the things that activate the acinar cells in order to secrete these pancreatic enzymes? Previously, we said the S cells will stimulate, the S cells will secrete secretin, and secretin will activate the ductal cells. But what about the acinar cells? Acinar cells have both CCK and acetylcholine. And from where they are secreted from a cell called I cell. We will come to this concept again, and I will explain it more. Let's move on. This is a pancreatic secretion. This graph shows a pancreatic secretions when there is absence of food. The left side is absence of food, which is no food. And the right side, or the right graph, is when you eat food. So let's see here, if you notice, there is a spikes on the left side when there is no food. And why is that happening exactly? Because simply in your body, there is a rhythm, like every 90 to 30 minutes, the body will start to make the GIT, the gastrointestinal tract, move and secrete enzymes. And this is called a rhythm. But when you have a meal, which is at 0 hour, you will see that the enzymes are being spiked and they are going outside. Here in this graph, they are talking about the amylase, but this is the same concept for every pancreatic enzyme. And don't worry about the low carb diet and high carb diet. Just know the difference in absence of food and presence of food. Yeah, so let's revise one more time the enzymes and the cells. So we said, let's look here at the secretin. What's the cell that releases the secretin is the S cells. The S cells will go secrete secretin, and when secretin go bind to the receptors in the ductal cell, it will release cyclic AMP. And from cyclic AMP, it will go and release bicarbonate. However, in the acinar cells, we have CCK, which comes from the I cells. And when I cells is releasing the CCK will go and it will not activate cyclic AMP this time. It will release the calcium. Tamam? Know the calcium and know the cyclic AMP. This is physiology. They love to ask this question, even though it doesn't make any sense. So the calcium is released after the CCK binds to the receptor. Okay, and after the calcium, it will go and release the pancreatic enzymes. I hope this concept is clear, but don't worry, we will come again to this concept in order to make it more. This is a checkpoint. Do you have any question for anything previously? Or do you want me to clarify anything or say something again? or should we move on? All clear? Good, alhamdulillah.
Clear.
Okay, thank you. Now, regulation of pancreatic secretion. What does that mean? It means, what is the thing that really tells the pancreas to secrete the juice. Juice means both bicarbonate fluids and pancreatic enzymes. So what is the main thing that tells the pancreas let's secrete some juices? We have three main phases. The first one is cephalic. What does cephalic mean? In medicine, or in Greek, cephalic means head. So when you smell a food or when you see a food, the pancreatic secretions will start, or the pancreas will start secreting the juices. But however, it's only a small portion. It's not even significant. It's the same thing for the gastric. It's not gastric. It's gastric phase. It will not be significant. However, when the food reaches to the small intestine and the duodenum, the pancreas will start secreting a lot of or a significant amount of pancreatic juices. And why do you think, guys, the intestinal phase is the more significant one? Just think out of the box. Why do you guys think the pancreas secretes the significant amount of enzyme when the food reaches the intestine? Link the things with the intestine. Okay, because it's when digestion is ending. You're close. And someone is saying fat absorption due to bile and the secretion that drain into the saliva via ample of water. Okay. Yes, I got half of the answers, mashallah. And Sidra also. Like, the reason behind that is you have the S cells and the I cells in the small intestine and the duodenum. And what is the things that stimulate the pancreas is S cells and I cells. So basically, the I cells and the S cells have different stimulus. For S cells, the hydrogen or the acid environment will stimulate the S cells. However, in the I cells, it's food, which is fat, and proteins and sugars. So you guys got the answer? MashaAllah. Right, let's go to the next part. This is a game. We said the S cells will be activated by the hydrogen or the acidic environment. And when the S cells is activated, it will release what? It will release secretin. Secretin will go by cyclic A and B and release bicarbonate in the ductal cells here. On the other hand, what stimulates I cells to secrete CCK? It's different stimulus. This time it's not acid or hydrogen. This time, phenylalanine, methionine, and tryptophan, which is amino acids, and also small peptides, and also fatty acid will stimulate the I cells. Fatty acid is basically lipids. Small peptides is protein. Amino acid is also protein. So remember, fatty acid and proteins are the one who activate the I cells. The most potent one is fat. and then protein, and less likely the sugars or the carbohydrate. So when these things, the fat and protein, activate the eye cells will secrete CCK. And the CCK will go and secrete calcium by a signaling pathway, and the calcium will go and secrete all the enzymes that we need from the acinar cells. And this is again the same concept. I repeated it a lot to make you guys understand it and memorize it. Again, S cells from the duodenum will receive hydrogen, and then it will release secretin, and secretin will go to the ductal cells. Why ductal cells? Because ductal cells have the bicarbonate. On the other hand, when we receive our proteins and fat, this protein and fat will activate the eye cells. The eye cells will release CCK and sometimes acetylcholine, but just remember CCK. CCK will go and bind to the acinar cells, and the acinar cells will go and secrete pancreatic enzymes, which is responsible for digestion of all the foods or all the energy sources. Like, how does the CCK go to the pancreas and tells the pancreas, here is a CCK, yalla, release some enzymes? CCK have two pathways, and it's important to know them. Doctor might ask about one of them, okay? The first one is the blood. So the eye cells and the duodenum will secrete the CCK to the veins, and from the veins it will go to the arteries, and from arteries it will go back to the pancreas. And it will go to which receptor and which cells? It will go to the acinar cells. And then the acinar cells will secrete it. And there is another pathway which is more important, and doctor even might ask about this, is called the anterior pancreatic reflex or vagovagal pathway. Okay, this pathway it does not utilize and uses arteries and veins. This time, the CCK will try to go into the neurons. So basically, when the CCK go, it will bind to the vagal afferent. When we say afferent, it means a neuron going to the brain, okay? So the vagal afferent will take the CCK and go to the brainstem and the dorsal vagal complex, and the dorsal vagal complex will send a signal pathway through the efferent, through the vagal efferent. Efferent means from the brain to the part of the bodies. And this vagal efferent will release acetylcholine and VIP. And acetylcholine will go and secrete to the acinar cells, and acinar cells will secrete the pancreatic enzymes. Again, simply, CCK will go into the vagal afferent, which is going to the brain, go to the brainstem here actually, and then it will go back by vagal efferent, and from vagal efferent it will release acetylcholine and VIP. And I think finally we have what's the most important thing, or what's the potent activator or stimulator for CCK. Remember when we said fatty acids? So the first and the most potent stimulator is the fat or fatty acids for activating the CCK and releasing CCK. The second most one is a protein, which we said small peptides and amino acids, and the least one is sugars or carbohydrates. So first is the most potent is fat, then protein, then carbs. If you don't know يعني what I'm talking about, I'm talking about this one, which is amino acids, then small peptides, then fatty acids will go and release CCK. Okay, the final slide of today. Pancreatitis. Do you guys remember when I said the gallbladder, which is here, shares the same opening with the pancreas? Do you remember this, guys? I hope you remember this. It was at the beginning of the lecture. طيب. The thing is, when there is a stone in the gallbladder, it might stuck in 3 locations. But the location that I'm concerned with is the ampulla of water. So when there is obstruction in this area from a stone from the gallbladder, it will go and stuck in the ampulla of water, which will cause a retention of the enzymatic pancreatic enzymes. So the pancreas will try to push some enzymes into the duodenum, but the pancreas cannot because there is obstruction. So what happens here is now, because of the obstruction, the enzymes will go back and it will start digesting itself. This is called pancreatitis, or another way or other term, this is autodigestion. So how can I know this person is having a pancreatitis? Simply by seeing the symptoms. The pancreatitis people or patient will have 4 symptoms. The first one is steatorrhea and diarrhea and malabsorption and abdominal discomfort. Okay, so what does steatorrhea mean? Steatorrhea means you are seeing a lipid or there is lipids in the feces. And why is that? Because there is no pancreatic secretions going to the duodenum. So there is no breaking down of lipids. Thus, there is no digestion of lipid. So the lipids will go all the way from the duodenum to the large intestine without being absorbed because it's not breaking down to simpler form or simpler form. And then diarrhea and malabsorption, of course, because there's no enzymes that can digest the food that's coming from the duodenum, and abdominal discomfort because pancreatitis is inflammation of the pancreas. If we put titus after any word, it means there's infection. And don't really worry about this one, which is the causes of the pancreatitis. And you just know the concept. The doctor did not really go through it, so I don't think the causes is important. Maybe just know the goldstone example that I gave you. So I think, guys, we're finished. I'll just give you a summary, then we go to the quiz and practice questions. So we said pancreas secretes isotonic fluid. What does isotonic fluid mean? It means the sodium, potassium, and ions will be the same as the plasma. Okay? It will secrete what? The pancreas will secrete what? Will secrete bicarbonate and will secrete also enzymes. Bicarbonate have 2 functions. It will neutralize the acid, and because it's a base, and it will activate enzymes, which is pancreatic enzymes. Why is that? Because remember, the pancreatic enzymes have the peak function in alkaline environment, which is opposite to the stomach. Just remember, stomach acid, pancreas is more alkaline. For the bicarbonate, it's released from the duct cells. And what is the thing that stimulated is secretin. From which cells? From the S cells. From where? From the duodenum. Okay, and the chloride, and there is two main excretion types for bicarbonate, which is bicarbonate chloride exchanger and CFTR. Okay, and when we have low flow rate, the chloride will be more, and when we have high flow rate, the bicarbonate will be more. And for the enzymes, just quickly, it's a zymogen or precursor, which means inactive form, and they are released from the acinar cells. And the main stimulus is CCK. From which cells? From I cells. From where? From the duodenum. And the most potent stimulus for CCK is fat, then protein, and less likely is sugar or carbohydrates. And what activate the CCK, or what CCK will activate what? It will activate the enteropancreatic system, which is vagal, and will go by blood also. And the diseases we discussed, the pancreatitis. We are done with the lecture. Do you guys have any questions? I'll be more than happy to answer you. Do you guys have one? Very clear, thank you. Okay, we have a quiz now. Feel free to, you can unmute yourself and answer and read the questions, or you can just read the questions and type the answer in the chat. Okay. I'll give you 1 minute and you can answer in the chat if you want. Happy guys, make mistakes now. Don't make it in the exam. We got 1A. What about the rest? I think A. Okay, let's read the question. Which of the following hormones is produced in the duodenum? So first of all, it should be a hormone or enzyme from the duodenum in response to digestion. The second thing, it should be response to digestion product and stimulate the pancreas, which is also, the third one is stimulate the pancreas in true hormonal fashion to produce enzyme-rich secretion. And it should be an enzyme. Is CCK matches all these? So, is it released from the duodenum? Yes. Is it response to digestion product? Yes, which is fat and proteins. Does it stimulate the pancreas? Yes, it does, by two pathways, which is blood and vagal. Does it produce an enzyme-rich secretion? Yes, it releases all the enzymes. So the first question is A. Bravo, guys. The second question. Yalla, 1 more minute. This question is kind of hard. I got E, E, E. Okay, let's read the question. How does secretin stimulate the secretion of bicarbonate from the pancreas? So simply, he's asking what's the function of secretin, or how does the secretin act on the ductal cells to release the bicarbonate? Does it close the potassium channel? I don't think we studied potassium channel. Does it close sodium channels? Same thing. We did not study sodium channels. Does it decrease nephrons level? We did not study we did not study this. Increase acetylcholine level? No, it does not. It will stimulate, or it will open CFTR channel. CFTR channel is responsible for chloride. So the answer is A. So bravo guys again. MashaAllah. It's becoming harder and harder. Third question. One more minute. We have one thing. C question mark. Come on, guys, be confident. Okay, Allah, let's read the question. Let's read the question. As a part of large B, D, and E are incorrect. So leaves with A and C. Okay, let's see. As part of a large grant studying pancreatic adenocarcinoma, if you don't know what's adenocarcinoma, it's cancer in the glands. Baseline data of pancreatic exocrine secretion are obtained from a healthy people. So basically, they are asking about pancreatic exocrine secretion. Okay. A. Its enzyme concentration is increased with pancreatic polypeptide. No, it's wrong. Wait. B. Its bicarbonate concentration increases with increasing chloride concentration. Is that true? No, it's not true, because bicarbonate and chloride are always opposite to each other. When the bicarbonate increases, the chloride will go down. Okay. C. Its bicarbonate concentration, which increases with the secretin stimulation. So is that correct? Yes, it's correct. We said the secretins will stimulate the bicarbonate to be secreted out. So C is correct. Don't worry about DNA, it's wrong. Yeah, C. Final question for this debate. We have 1A question mark. And mashallah, almost all your answers is question mark. Okay, C question mark. Okay. Yalla, let's read the question. 2-month-old baby present with vomiting, diarrhea, and failure to thrive. On investigation, we have edema and ascites are found. And blood values show hypoproteinemia. Okay. Several attempts with different protein diets are unsuccessful, but the administration of the pancreatic enzyme alleviates the symptoms. So basically, they are saying he cannot digest the proteins. What's the problem? Either C or A. Either C or A. Okay, but probably since anterior peptide is responsible to the conversion of inactivate to inactivate, so E. Wait, C. Like, the answer is C. Why is that? Because, as Jasser said, anterior peptidase is responsible for conversion of trypsinogen to trypsin. Okay. Yes, I think we reached to the conclusion for today. Thank you, guys. I hope I would appreciate if you saw this. And if you have any question, feel free to ask now or later on WhatsApp. And you have a good day. Have a great day, guys. Thank you. Thank you so much.
Thank you so much.
Have fun. Yeah, I hope to see you guys again.